start. Hello everyone. So today we will hear the PhD dissertation talk of Sutapa Patra, supervised by Alon Omrani and Yoav Rosenberg. You want to say a few words?
כן, שלום לכולם, So Sutapa, indeed, she's about to finish her PhD. A couple of words. About 10 years ago, we had the privilege to put our hands on a 9-meter core that tells a very interesting story about the KPG mass extinction. In a very preliminary work, we managed to measure some bug proxies, and we understood at that point that we had a very interesting story that we wish to understand and tell. And we were looking for a PhD student, and Sutapa arrived from India. And through the cultural differences and the different language outside of course the working space, she managed to work hard and to produce a molecular work, which is never easy as a topic work. And hopefully she will finish this with a couple of publication. And here she will concentrate on a certain part of it. And I hope you will enjoy the story and congratulate Sutapa for finishing a very serious PhD.
Thank you all for the nice introduction, and thank you everyone for coming. So good morning. So today I am going to present my PhD thesis work, and the title of my thesis is Geochemical Characterization of Primary Producers and Organic Matter Preservation following the Cretaceous-Paleogene mass extinction at the southern Tethys upwelling ecosystem. So I have started my PhD since March 2020 at the Hebrew University of Jerusalem with Professor Alon Omrani and Dr. Yoav Rosenberg in Geological Survey of Israel. So for the next 45 minutes, I am going to cover these following things: a little bit of background of my work, the objective, methods and instrumentation, and some important results of my work and conclusions. So I would like to start my talk. What is extinction? Extinction is a fact of evolution, and throughout the Earth geological history, there are big 5 major mass extinction events happen. One of the biggest and well-studied mass extinction is the Cretaceous-Paleogene mass extinction. It occurred around 66 million years ago, and now all of us are quite familiar about this period because dinosaurs went extinct. And it was thought that there could be a large meteorite impact that caused this mass extinction. However, there is another school of thought where it considered that could be that Deccan flood volcanism was also a responsible factor for this mass extinction. However, in today's talk, I am not going to debate over what is the reason of this mass extinction, rather the effect of the mass extinction. So the effect of KPG mass extinction has a devastating. It causes so many perturbation in the ecosystem as well as the environment, such as sulfate aerosols fill the atmosphere and covered a blanket. As a result, there is a sunlight become blocked and reduced in the photosynthesis. Ocean become anoxic. And also there is in this situation 93% of species went extinct because they could not adapt to the severe changes. Also this marine life suffered a huge loss. 85% of calcareous haptophyte algae, that the building block of the primary producers, went extinct in this KPG mass extinction period. So this is a very conceptual framework of a marine ecosystem. So as you can see, this is divided into 3 parts, like euphotic zone, the water column, sediments, the euphotic part where it catches the sunlight, so there would be abundance of life. And this is the sinking of the organic matter, the export productivity, and eventually this organic matter breakdown in the sediment and recorded it as a TOC. What happened in the scenario of high productive marine ecosystem? So it would be increased in the total organic matter on the surface, and as a result there would be increase in the export productivity. So the bottom water eventually become very anoxic, and in that anoxic zone there would be some microbial communities who are using sulfate instead of oxygen for their microbial respiration, and reduce and release hydrogen sulfide into the sediment. This reduced sulfur species eventually bind with the iron to form pyrite, as well as it will bind with the functional sites of the organic molecule to form organosulfur compounds. These are the two framework of a marine ecosystem and the high productive ecosystem. Now, whenever if there is a perturbation like a big event KPG, this system will be changed, and it can affect in various way, like all these functional properties and the community structures would become affected. So, as the community structures means the primary producers affected by the KPG mass extinction, and the recovery of primary producers varied across different marine ecosystems, and it eventually generates several hypotheses. So one is the Strangelove ocean hypothesis, where it was assumed that there is a almost complete reduction of primary productivity for a prolonged period of time, like up to several million years. So you think this is a pre-extinction scenario of a marine, I would say a healthy ecosystem, where there is a lot of primary producers on the surface like phytoplankton, algae, bacteria, and is a sinking of organic matter flux due to good export productivity, and eventually the breakdown of organic matter gives a high TOC. Now, when in this Strangelove ocean hypothesis, where it was considered that almost complete cessation of primary productivity, so very less amount of organic matter would be eventually recorded into the sediment. So another hypothesis is the living ocean hypothesis, where it was considered that the surface ocean was alive, but the biological pump get lost. So eventually there is a reduction of the export productivity, and and less amount of TOC would be eventually recorded in the sediment. And the hypothesis tells that this situation continued up from 1,000 years up to 3 million years. So both of these two hypotheses are generally based on the fossilized skeletons of organisms. Later, with new scientific advancement, tools and techniques, a new hypothesis came into the scenario, which is known as heterogeneous ocean hypothesis. In that case, it was considered that the recovery of primary producers varied across different marine ecosystems, and it's more rapid, like less than 10 to 30, within the 10 to 30 kilo years. So all these are three main hypotheses, and these two hypotheses, Strangelove ocean and living ocean, the assumption was made for global decline, while in case of heterogeneous ocean hypothesis, it was considered that it does not reflect a global trend, but it more depend on the local factors. Now, local factors mean it mainly depend on the components of the ecosystem as well as the local environmental conditions. So, ecosystem has two components, ecosystem function and ecosystem structure. These two terms I have adopted from the paper Hall and Tarok 2013. What is ecosystem? The productivity, preservation, nutrient cycling, all these physical properties, that means the biophysical properties that shape the ecosystem function. While the microbial species, the community structure, the species distribution and the trophic interactions that shape the ecosystem structure. So any kind of external disturbance can affect the ecosystem function as well as ecosystem structure, or their mutual interactions can be disrupted. So in my study, I will look how these two components of the ecosystem, like ecosystem function and ecosystem structures, get affected by the KPG mass extinction. So there are several open questions related to KPG. So the main three important questions that I am trying to look in my study in the paleo-environmental conditions, how the ecosystem function shaped in the upwelling Tethys belt before and after the KPG mass extinction, how the community structure, that means the primary producers, response in the upwelling Tethys belt before and after this KPG event. And in the third question, that how did the response of upwelling ecosystem differ from other marine ecosystem across the world. So the previous work that is done by Rosenberg et al. 2021 from the southern Tethys upwelling belt, the section is from the Golan, and he analyzed a 6-meter long core, and this is a stratigraphic profile, and I am going to show you more profile like this. And this biostratigraphy spanning from P higher to P2 and also the age between Cretaceous and Paleogene. This red mark indicates the boundary between the pre-extinction and the post-extinction level. So in this study of Rosenberg et al. 2021, where he used the bulk geochemical proxies like productivity, and he found that the productivity remained constant throughout the extinction period, especially up to this point, like more than 140 kilo years. So this is the raw image of the core section, where you can see that the rock is very dark in color, exploded with organic matter, and there is no color change in the pre and the post extinction levels. So the TOC was kind of continuous in that section. So the, Rosenberg et al. 2021 concluded that there is a resilience and stability across the KPG. But what about the primary producers? What about the non-fossilized microorganisms? So these actually give me the motivation to further look into the deep detail at a molecular level to see the changes in the ecosystem function as well as in the community structure. So this is the paleo map that showing southern Tethys upwelling ecosystem, which also follow this heterogeneous ocean hypothesis model. And now you have noticed that I am studying an upwelling ecosystem. So I would like to show you how this upwelling ecosystem is different from other two marine ecosystems like neritic and bathyal. So the neritic ecosystem that lies on the top of the continental shelf from the sea surface to 200-meter bottom of the ocean, where it can get a lot of sunlight, it's abundant with life, lot of phytoplankton, zooplankton. On the other hand, the bathyal ecosystem, where it starts from the 200-meter of the bottom of the ocean to 6,000 meter, and where the ocean is very dark, deep, sunlight cannot easily penetrate there, and life is in very harsh condition. So interestingly, the upwelling ecosystem, which lies on the top of the continental shelf, but due to Coriolis force and the wind transport, the cold nutrient-rich bottom water come to the surface and make the system very high productive. As you can see from this table, that upwelling ecosystem only consists 0.1% of the total oceanic area, but it has a very high annual productivity, 250 to 350 gram carbon per meter square per year. So the main research objective to my study is to develop a mechanistic understanding of how an upwelling ecosystem responds to an ecological crisis such as KPG mass extinction by assessing the ecosystem function and community structure proxies in a by using molecular biomarker proxies and their associated isotopic signatures. So my thesis is divided into 3 chapters, and each chapter consists one goal. So the first chapter, I have analyzed the molecular evidences of ecosystem function and community structure proxies across the KPG. In chapter 2, insights into the organic sulfur cycle from both bulk and compound specific proxies in the context of the KPG boundary, and in the third chapter, the comparison the effect of KPG mass extinction between ecosystem function and community structure proxies across different marine ecosystems. So the research approach and method. So in my study, I have mainly used the tools biomarkers. So what is biomarkers? Biomarkers are biological derived organisms, and it is also known as molecular fossils or lipid fossils. So be it eukaryotic or prokaryotic cell, there is a component in the cell membrane known as lipid, which is less prone to degradation, and as a result, even the organism die, the information about this organism recorded into the sediment. So let's see an example. The eukaryotic cell has a cell membrane component known as sterol, and when the organisms die, eventually it buried into the sediment, and upon diagenesis with pressure, temperature conditions, the sterol converted into the geomolecule known as sterane. Similarly, in case of prokaryotic organisms like bacteria, they have the cell membrane component known as hopanol, and upon sedimentation diagenesis, it will be converted into hopane. So if we take the ratio between sterane and hopane, it eventually gives the information between the eukaryotes versus prokaryotes contribution. In this way, the biomarkers give a tremendous amount of information about the depositional history and the microbial communities of the ecosystem. So this sterane and hopane is an important proxy for biomarkers, and throughout my study I have analyzed many more proxies like this, which I am showing in my results section. So the molecular proxies that I have used for the ecosystem function, the bulk proxies for productivity like TOC and CaCO3, that means total organic carbon and calcium carbonate, nutrient cycling like carbon by nitrogen organic ratio and the nitrogen isotopes, preservation, the molecular proxy pristine phytin for anoxic condition, and isoreneratin, the molecular proxy for euxenic condition. So what is euxenic condition? So let's see this diagram. So I have already shown you a very simple diagram in one of my introductory slide, that when there is a microbial sulfate reduction process in the anoxic zone of the sediment, these organisms use the sulfate for their microbial respiration and release the hydrogen sulfide into the sediment. Now if the microbial sulfate reduction process is too high in the sediment, eventually there would be lot of H2S generated into the sediment, and it started mixing into the water column. So as a result, the H2S started moving into the upper water column, and in that zone where it will still get sunlight, but it is poisonous with the presence of H2S concentration, in that zone there are some bacteria started living known as green sulfur and purple sulfur bacteria. They are the biomarkers that derive from these organisms as known as isoreneratin. So the presence of isoreneratin is a very good proxy to understand the photic zone euxenic condition. And also I have used the gamma serine index for stratification, and bulk organosulfur and pyrite and their isotopes as a preservation proxies, as well as the ecosystem function. The community structure, that means the ecosystem structure proxies that I have used in my study, C27 sterane, that means the marine algae, C28 sterane, nanoplankton such as diatoms, coccolithophores, dinoflagellates, C29 sterane like green algae, and the hopanes for bacteria, and photic zone euxenic biomarkers like green and the purple sulfur bacteria. So this is the raw image of the core section that I have already shown you, and you can see that the core is spanning from 1036 to 1039. It's a 3-meter core, and the biomarker biostratigraphy is P hat to P1C, and it's showing a dark, homogeneous, organic rich calcareous sediments with some fine laminations. And this white color, this white star symbolizes from where the samples I have analyzed for my study. And this boundary is between the Cretaceous and Paleogene. And if you look carefully, that I have tried to collect most of my samples close to the boundary, so that the effect of the KPG mass extinction would be reflected in the both pre and the post extinction level. So you can see from this diagram that total organic matter consists a very small portion of the total rock, and the total organic matter has two components. One is the large insoluble macromolecular fraction, which is known as kerogen, while the soluble fraction is known as bitumen. Bitumen consists a very small part of the total organic matter, and bitumen has two components, asphaltene and the maltene. The maltene consists saturated aromatic and polar hydrocarbons. So I have analyzed most of my samples from this saturated, this maltene fraction, the saturated aromatic and the polar fraction. So after doing the lipid extraction and the separation with the liquid chromatography, the saturated aromatic and the sulfur-rich fractions are analyzed into gas chromatography mass spectrometry to identify the biomarkers like pristine, phytane, styrene, hopane, etc. and to determine the delta 13C value of each of the hydrocarbon compound. We used compound specific carbon isotope analysis with the GC-IRMS, and also to determine the delta 34S values of each of the organosulfur compounds at picomole level. I have used GC-MC ICP-MS, where a GC is coupled with the multi-collector inductively coupled plasma mass spectrometry. So now I am going to show you the important results of my work. So the bulk proxies from the ecosystem function. So I have analyzed a almost 2.2 meter core section, and that's spanning between Cretaceous and the Paleogene period. This is the very thin P0 boundary, and this red mark indicates the boundary between the Cretaceous and the Paleogene section, between the Cretaceous and Paleogene. So you can see that the total organic carbon is almost constant throughout this extinction period, especially up to this level, that is kind of 140 kilo years after the post-extinction interval. And similarly, the total sulfur amount, which is also remain constant throughout the extinction boundary, which is greater than 1, that also means the system is very anoxic. Similarly, the kerogen sulfur and the pyrite sulfur, which is also remain constant throughout the extinction boundary, and also the kerogen sulfur is much more dominated compared to the pyrite sulfur. Now the isotopes between this kerogen sulfur and the pyrite sulfur, they are not showing much changes across this boundary, although the resolution is not good here, but you can see that the delta 34S of the pyrite is almost 20 per mil depleted compared to the bulk kerogen sulfur. Why it is like this? It's due to the preservation process that enhanced by the secondary sulfurization driven by the reduced sulfur species generated through microbial sulfate reduction process. Let's see a diagram how these things are happening. So in the surface there is already organic matter, but in a high productive system there would be a lot of that means increased of organic matter input. So eventually the export productivity would be increased and the bottom water become very anoxic. And now that anoxic zone, which I have already explained in my previous slides, that microbial sulfate reduction process going on, and they are using this sulfate for their microbial respiration and release this hydrogen sulfide, and this reduced sulfur species eventually bind with the iron to form pyrite, and this process is known as microbial sulfate reduction process, which eventually gives a very large isotopic fractionation that lies from -20 to -75 per mil. That you can see that the pyrite, that's why is too much depleted, while also that if there is a lot of organic matter in the sediment, this reduced sulfur species eventually bind with the functional sites of the organic matter to form organosulfur compounds. So from this result I can conclude that there is a continuous productivity and preservation conditions across the KPG interval. That means the ecosystem function was kind of resilient throughout the KPG boundary. Let's see how the molecular proxies behave. The anoxic proxy, pristine phytin, which is less than 1, that means is very anoxic throughout this extinction profile. And similarly, the reducing conditions, the proxy for homohope index is also remain constant throughout the extinction profile. And the stratification proxy, the gamma serid index, which is greater than 1, that means the water column was highly stratified. So if we look at this figure, you can see that how this stratification developed, and so the conclusion from this result is that continuous anoxic and reducing conditions that persistent throughout this KPG interval, and there is a persistent water column stratification across this KPG boundary. So now I would like to show you how this consistent anoxic conditions, so the situation is not only anoxic in my system, it's also euxenic. How? By the presence of this isoreneratin and their derivative of aryl isoprenoid. As you can see, they are present before the extinction and also after the extinction. So, but during the post KPG interval, there is an increment in this isoreneratin, that means euxenic conditions increased in the post KPG interval. But this euxenic condition is not constant throughout the extinction period. Rather, it was episodic in nature. As you can see that I have used the proxy for that, aryl isoprenoid ratio. Now, if I look at this diagram, that due to this microbial sulfate reduction process, lot of hydrogen sulfide started mixing into the water column. And in that water column, as a result, the chemocline of the water column started rising. And in that zone, where it will still get sunlight, but it is full of hydrogen sulfide concentration, there are some bacteria known as green sulfur and purple sulfur bacteria. They are started using this H2S for their own microbial respiration, and the biomarkers that derived from these organisms is known as isoreneratin and aryl isoprenoids. So, the presence of this photic zone, this green sulfur and purple sulfur bacteria eventually tell us the system is highly euxenic throughout the extinction period. So, my conclusion from this result is that there is a continuous presence of euxenic condition prior to the KPG event, and there is a temporal increase in the photic zone euxenic during the transition interval. So, now I would like to show you how this continuous anoxia and euxenia condition affected the community structure proxies. The general source indicators from the community structures are derived by analyzing the N-alkane proxies. So, as you can see that after the KPG up to like 140 kiloyears, the system is mainly dominated by these short chain N-alkanes. That means it is coming from the microbial organisms like phytoplankton, algae, bacteria. But the high molecular weight N-alkanes, which is a proxy for the terrestrial organic matter, is not very significant. It's even, the scale is even less than 0.5. So, the overall the system is not, there is a less contribution from terrestrial organic matter. However, there is an immediate increase just after the extinction in the terrestrial organic matter. Further support of these two proxies, also I have analyzed the nitrogen isotopes. So, the atomic carbon by nitrogen ratio, which is like very high values, range between 35 to 40 throughout the extinction profile, and that means the marine organic matter deposited in a very high productive region where water column anoxia persists. Under such conditions, the microbial degradation enhanced denitrification, and they are using the nitrogen rich amino acid, and eventually the sediment is retained with the high values of carbon by nitrogen ratios. Further support of this data, the nitrogen isotope, which is kind of typical 5 per mil, and it's also a slight decreasing towards the post KPG interval like 1.5 per mil. It means that there is some denitrifying organisms, probably some heterotrophic bacteria that are functioning in the denitrification cycle. So if you look at this scheme, that this is the denitrification process going in the water column. So the main conclusion from these general source indicators that there is an overall contribution from the marine microbial communities, and there is a transient increase in the terrestrial organic matter input immediate after the KPG. And there is a high productivity that eventually led to denitrification within the water column, and the selective consumption due to the bacterial, the nitrogen rich amino acids that increase the carbon by nitrogen ratio into the sediment. Now going to the more source specific proxies for the primary producers, the building block of the food chain, I have used the proxy like stearine. So no matter what kind of stearine I am looking at, but it's remained kind of constant throughout the extinction profile. Similarly, the nanoplanktons, the proxy between nanoplankton versus green algae, that means the known as C28 by C29 stearine, that is the relative distribution between the diatoms, coprolithophores versus green algae, it also do not show any significant change across this boundary. Similarly, the relative distribution between prokaryotes by eukaryotes, which I have shown by this stearine by Hoppen, it's also remained constant throughout the extinction profile. So you can see that even before and after extinction, the community structure remained resilient. So there is a less significant change across the relative distribution of primary producer proxies, and the primary produce, that means that primary producers thrived well across the KPG. That means even there is a large catastrophic event of KPG, the photosynthesis continued. So, so throughout my proxies, I have shown you that most of the cases it remained constant or resilient, but there are 3 proxies that I have found show some distinct changes immediate after the KPG, although these changes are very transient. For example, this high molecular N alkanes, the proxy for the terrestrial organic matter, showing a transient increase immediate after this KPG interval, and also the aryl isoprenoid ratio, which is a proxy for the eugenic conditions like persistent versus episodic, and it's showing a severe eugenic condition after this KPG event. And the nitrogen isotope, which also show a relative decreasement like 1 per mil towards this post KPG interval. So here I am trying to understand if there is any control on this microbial biogeochemical process. So I have tried to make a correlation, and you can see that there is a nice correlation, that square is 0.7 in high molecular weight N-alkane versus aryl isoprenoid ratio. So as you can see that when there is a lot of terrestrial organic matter input in the sediment, eventually the eugenic condition increased. That's why it's reflecting from this plot that with more terrestrial input there is a severe eugenic conditions. Similarly, there is also the delta 15N, the nitrogen isotope has a control over this terrestrial input, that means that terrestrial input high and there is a reduction in the nitrogen isotopes. As you can see, with the high terrestrial matter input, the nitrogen isotope is decreasing. Also it's showing a good correlation. So I hypothesize that enhanced influx of organic matter into the ocean likely contributed to the development of more anoxic conditions. So to this end, I have conducted a comprehensive comparison of upwelling Golan along with other marine ecosystems that experienced KPG by utilizing published literature datasets to identify a recovery pattern in both ecosystem function and community structure proxies. While most of the studies in the post-KPG interval focused on the duration of the recovery, but there are very few studies who are trying to understand the recovery pattern in the post-extinction interval, but there is no definitive conclusions has made. So in my study, I am trying to identify a recovery pattern in both ecosystem function and community structure proxies. These are the study sites that I have incorporated in my comprehensive comparison, and this is the paleomap of upwelling Golan with other marine ecosystem. So the comparison I have done in both ecosystem function and the community structure. To facilitate the comparison, I have chosen two proxies, total organic carbon and calcium carbonate, the proxy for the productivity, and look at their changes in their post to pre-extinction level. And it shows that most of the ecosystem decreased in calcium carbonate in their post-extinction interval, while the fish clay ecosystem stands out and there is an increment in their TOC post to pre-extinction level, and that reach up to factor 40. These ecosystems probably reflect that there is a decrease in the surface water productivity, but increase in the bottom water anoxic conditions that increase the TOC. But interestingly, the data from the Golan, it clustered near this unity ratio, and it means that there is a less significant change, or is very stable in both TOC and the CaCO3 post to pre-extinction level in this equivalent period. Now again, coming to the community structure proxies, I have chosen two parameters. One is for Sterane by Hoppen, that is the relative distribution between eukaryotes versus prokaryotes, and C28 by C29, the relative distribution between nanoplankton versus green algae. So this x-axis means, the right hand side means more contribution from eukaryotic communities, and the y-axis in the top side means lot of contribution from nanoplankton communities. Unfortunately, except Golan, we do only have 4 other ecosystems like fish clay, Agost, Mid-Waipara and Gulf coastal plain. We don't have much published data sets that have used these two proxies. And for each ecosystem, I have used a different symbol, and I have used a color code for the pre-extinction level. I have used yellow, P0 for red, and green for post-extinction level. Now you can see that it shows a kind of bimodal distribution pattern, and the ecosystem Agost and the Mid-Waipara, they are showing a larger variability in their nanoplankton communities, while Agost is showing a reduction in the nanoplankton communities in the post-extinction level. On the other hand, the fish clay and the Gulf coastal plain that lies on the x-axis of this plot and show larger variation in their eukaryotes versus prokaryotes contribution. And among all these ecosystems, upwelling Golan, it show a very less distribution in their Sterane by Hoppen proxies compared to all other ecosystems. So basically, after comparing with all other ecosystems, it showed that upwelling Golan showing a smallest amplitude in both ecosystem function and community structure proxies, and it remained remarkably stable throughout this extinction period. So the conclusion from this comparison is that a bimodal distribution pattern has observed in the community structure proxies, and unlike all other ecosystems, Golan is not showing larger variation in their pre or the post-extinction level. Rather, upwelling Golan remained relatively stable compared to other marine ecosystems. So what we have learned from this throughout these results, that in case of Golan, the ecosystem function proxies like productivity, preservation, anoxia, euxenia remain resilient throughout the KPG, and the relative distribution for the primary producer proxies that remain unchanged, and photosynthesis continued well across the KPG. So we can say that the devastating effect of KPG in upwelling Golan is seemingly unaffected. Now you can ask me the question, why is Golan is relatively stable? So here I have tried to put all the my observations together and try to understand in a conceptual way how this Golan is functioning. So as you can see that Golan is a high productive upwelling ecosystem. So there is a lot of organic matter on the surface, and eventually there would be a lot of export productivity, and the bottom water would be very anoxic in the sediment. So in that zone that I have already explained a couple of times that microbial communities living, they are using this sulfate for their respiration and releasing this hydrogen sulfide into the sediments. These microbial communities known as microbial sulfate reducers, and when this too much organic matter is coming to the bottom, this microbial sulfate reduction process enhanced. As a result, eventually this H2S started mixing into the water column, and in that zone where it still gets sunlight, but there are some bacteria, this green and the purple sulfur bacteria, they are using this hydrogen sulfide for their respiration and release this H2S as a form of sulfur or sulfate into the water column. These are also known as microbial sulfur oxidation. So you can see there is a two process going on. One is the microbial sulfate reduction and another is the microbial sulfur oxidation. So these two process of this reduction and the oxidation that helps the upwelling Golan to behave as a steady state, and it also makes the upper water column oxygenated to flourish the eukaryotic organisms. Apart from this microbial sulfate reduction and the oxidation, there are also some other functions. The denitrification is also going. So the conclusion is that due to this continuous cycle of this microbial sulfate reduction and the oxidation, the eutrophic upwelling in the Golan maintains a steady state, and which eventually supports flourishing of eukaryotic organisms and helps the upwelling Golan to survive external disturbances such as big event like KPG. So my final hypothesis is that the strong anoxic and euxenic conditions and the diverse bacterial communities that operated at various levels within the ecosystem enhanced the upwelling Golan ecosystem stability and made it resilient to external disturbances. So I'd like to thanks to both of my supervisors, Professor Alon Amrani and Dr. Noab Rosenberg, for their constant support. To all my lab mates, past and present, and to all the members from Organic Carbon Dynamics groups of Geological Survey of Israel, to my office colleagues, especially from the third floor, room number 314, and to all the staffs at the Institute of Earth Sciences at Huji, and to all my friends and family. And a special thanks I'd like to give to my mom, who is not present here today. And thank you for listening.
Thank you very much. Questions?
So, what you're showing is kind of strange, right? You're saying there's this huge mass extinction, right? That
Yes.
3/4 of fauna in the ocean basically would go extinct. And yet you don't see this change.
Yes.
So, the fact that you don't see the change, I think you proved very, very, in a very compelling way that there isn't a change. I want to ask about whether this is the mass extinction that you claim it is, right? So if you're in an upwelling zone, what is the sedimentation rate you expect in the ocean? An upwelling zone, which is not far from the ocean.
So the sedimentation rate for the upwelling Golan is kind of 4 to 3.6 meter per million year, and it was kind of resilient throughout this extinction period. And
so you're talking about, say, in your figure, it's 2 meters per million years.
yeah.
So that's a sedimentation rate that you get in the middle of the Pacific today, right? You don't get it near the shore in an upwelling region. The sedimentation rate should be 10 times that in that kind of time frame. Okay? And so this questions whether or not you're looking at what's happening in this time period is very interesting, right? Because if we believe the ages, which I want to ask also about how do you know what the ages are, you're seeing something which is very strange in a setting which is I would say very not typical to those kind of environments. Okay? And so that's, I think, it was more of a comment. Now the question, how do you know that you're in an upwelling region? And how do you know that this is actually the time period that you think you're looking at?
So the question for this upwelling, this is already, there are previous research on that the system is upwelling. It's not that I am saying, or there are many papers from the paleontological evidence, paleo-environmental condition, and it's well supported that the zone was upwelling.
But what is that evidence?
There are the community structure and the productivity, which is very high compared to other ecosystems, and also they have some other physical parameters which they have established quite well. Right now I could not exactly recall all the things, but it's not I am saying. And regarding this, why I am not showing changes, one problem could do, not a problem, the resolution that I am looking is, maybe I am going to back to the profile. It's like we have, this is 4 centimeter, we have difference in this period. So if we look at more finer resolution, maybe we can see a change, which I don't have this data right now. This one thing. And second thing, that if the system is very high productive, like the TOC is greater than 9%, if there is a small change, it would not be reflected in your study. So, but if the system is an organic matter lean, that means if the TOC is less, you can see a rapid change which is very significant. But in the system where it is already very high, even if there's something happened, it's not reflected in the sediment. That could be the one answer for this.
But based on what? The ages are determined?
The ages are determined by 2 ways. One is the Jarkon dating made for this Cretaceous-Paleogene boundary, and the biostratigraphy based on the foraminifera, which already published in Rosenberg et al. 2021. I have not analyzed the
What I noticed that if you leave it here, that many of your markers starts tens of centimeters before the boundary.
Yes.
Like this one, let's say diagram 15 or H and W, sigma and K. So what do you think is the reason that you see it? Do you see it also in the rock itself or just in the chemical?
No, I have seen all this analysis from the chemical way, from the extracting the information from the rock. And to do a molecular analysis, it's very hard to do like 1 by 1 centimeter. So I have chosen this resolution. So it's like compared to other studies, that this resolution will be fine to reflect the changes in the rock record, at least at a molecular level.
Okay, so I'm a little bit more curious about the oceanographic processes which controls the episodic unique conditions you have explained there, like whether it was controlled by the completely by the oceanography or by the tectonic or by the some volcanic eruptions happening there. What are the processes which control this?
Well, to give the answer that whether it's episodic or not, I didn't look at this oceanographic condition, but rather I would expect the changes in the paleo-environmental conditions. So if there is a more anoxic situation, so you will get more persistent in the nature. So the, let me show you. So this I have calculated based on the presence of this green and the purple sulfur bacteria and their abundance. And I have calculated the ratio between the high molecular weight and versus low molecular weight, and it shows that if the condition is oxic mode, there would be degradation in these larger carbon compounds. And so eventually it would be very high, but if there is a more anoxic conditions, these larger carbon compounds will be preserved more into the sediment. So again, coming back to your question, it's mostly depend on the preservation condition of the sediment. I'm not sure about the oceanographic condition, but of course the functional proxies, the preservation, that plays a huge role to say the system is eugenic or not.
And other one is like we talk about the terrestrial
Yes.
input. Yeah. So when we are talking about terrestrial input, it can be like a atmospheric, dust coming from the different places, or it can be a land derived material.
It's mostly from the land direct material, like the big trees, mostly from.
I'd like to follow up on that. So, which of those mechanisms that you talked about right at the beginning, whether you're talking about the impact of a meteorite or the Deccan flood basalts, which of those would be most likely to cause an influx of organic matter?
Well, that's a very good question. The recent studies mostly confirmed on the meteorite impact, and I would support the meteorite impact based on my results, because when there is a large thing coming to the atmosphere, so it makes the lot of sulfate particles coming into the atmosphere and then block the sunlight and everything, and eventually the ocean become anoxic. Those situations can be more prone to this meteorite impact effect based on the oceanic anoxic, because the organisms will die in this. Even the Deccan flood volcanisms, the recent study also show from this their carbon isotopes that it's not exactly crossing this KPG boundary. Its change is like 30 kilo years or something like that. So I would go mostly for the meteorite impact.
Does that mean that in the sea, while the sea ecosystems remained provably stable, the terrestrial systems didn't collapse?
No, all the systems actually collapsed, but here I am showing mostly from the marine ecosystem. But if you think that all of the things eventually dumped into the ocean, so there is a collapse, but the interesting thing of my study that my system is showing, okay, seems things remain pretty happy there, all the microbial communities.
Like you're a terrestrial, animal that lives at the upper...
They went extinct, all the large vertebrates, and there is also effect in the higher terrestrial communities, but this is not I am looking in my study. I am only focusing on the marine ecosystem.
So within the marine ecosystem, you showed that there are changes.
Very transient change, I would say. It's not very large or significant change.
Mm-hmm. So for instance, fauna that's living in the upper part of the water column would practically not be affected? Because
in other cases it affected, but here it shows, I've shown you that in the conceptual illustration that due to this continuous reduction and the oxidation cycle, that eventually make the upper water column oxygenated and helps the eukaryotic organisms, all these primary producers, to thrive well and remain stable in that system.
You said it, I'm sorry if I missed it. Is there an indication of the actual depth of the seabed? Like how deep was this region?
I have not taken the core and I don't have the information right now, but it's like 500 meter. Maybe Yoav can give the answer. Sorry, I just only focus on the molecular process. Maybe later I can give you the answer.
Okay, so thank you again.